So, welcome everybody to this webinar. It's really, as always, a pleasure to gather so many attendees for these webinars, which are an important component of the activity of the Global Virus Network. So, as always, a very few words about the Global Virus Network, and then we will move to the presentation from Hervé Perron. Global Virus Network, a network of now about 70 research centers and 12 affiliates all over the world, 3 missions: research with the task force, the watch groups, education and training with the GVN Academy, and advocacy and communication. A lot of partnership with industrials, with the corporate partnership program, which is very important, and many different activities you can find the list of on our website, and never hesitate to contact us for some details. And it is really in this context that we host these webinars, and today it's my pleasure, my great pleasure, to introduce Dr. Hervé Perron. Hervé has been trained, educated at the University Joseph Fourier in Grenoble in France, where he got his PhD, and then he moved to head to work in a joint research unit between a French research agency, CNRS, and bioMérieux. bioMérieux is a very large company focused on diagnostics and infectious disease. And then he moved to become the chief scientific officer of a company, which has been actually a spin-off of bioMérieux, and which is located both in Lyon, in France, and in Geneva, in Switzerland. And briefly, the contribution of Hervé have been very interesting and innovative findings of defining a novel family of human endogenous elements in patients with multiple sclerosis, but now there is evidence that it would be implicated actually in several autoimmune diseases. And recently have expanded this data in patients with COVID-19. So it's a very interesting and I would say original and innovative area, and we really look forward to this presentation. So Hervé, the floor is yours. Thank you so much.
Thank you. I would really like now to thank the Global Virus Network and Professor Christian Brechot for this opportunity to present our current research on the link between SARS-CoV-2 and the human endogenous retroviruses and its relevance to long COVID as a post-viral disease. So for the next 30 minutes, my presentation will cover the biology of human endogenous retroviruses, SARS-CoV-2 as a potent trigger of HERV-W envelope, HERV-W activation in COVID-19 patients, the continued HERV-W envelope expression in long COVID patients, and the need for precision medicine based on HERV-W presence in patients. So now, where do HERVs come from? These endogenous viral genetic elements come from ancestral infections with retroviruses that resulted in provirus retroinsertion into chromosomes of germ cells of an individual. And when the modified gametes did not cause lethality, retroviral genes were passed on to the offspring through heredity. However, repetition of such events with various environmental retroviruses and their intrinsic ability to retrotranspose also created variable recombination events, thereby yielding complex multi-copy HERV families with more or less inter-individual heterogeneity. So while physiological sorry. So while physiological genes only represent less than 3% of chromosomal DNA, the superfamily of transposable elements accounts for about 42%. And the human endogenous retroviruses, or HERVs, are parts of retrotransposable elements, and themselves, they represent about 8% of the human genome. They may be involved in various functions, are most often epigenetically silenced, less often retain protein coding capacity, or can remain inactive. I must complete the picture by illustrating the genomic heterogeneity among human individuals regarding HERV copies. This slide shows on the left the percentage of individuals from various geographical areas and ethnical origins carrying identified copies from the HERVCoV family. On the top lines, one can see dark blue color in all humans, indicating acquisition before human speciation. Then below, variable color intensity indicates a lower percentage of individuals with the corresponding HERV copy in their genome. Finally, some copies may be found in rare individuals and or in isolated ethnic groups. So on the right side, a recently discovered copy from the HERV-W family on chromosome 18 is also seen to be either absent with only solo LTR remains after an ancestral elimination of the proviral insert, or to retain the provirus sequence under hetero and monozygote chromosomal insertion. And this is variable in different areas in different populations. So the HERVs can be both beneficial or detrimental. And now, leaving apart the beneficial role of other HERV sequences and proteins, which have also been very well characterized, I will address the question of the activation of normally silent HERV copies causing pathogenic effects when they are activated or unsilenced. This slide just illustrates the numerous publications reporting the abnormal activation of the HERV-W family and the expression of its envelope protein, which have experimentally been induced by viruses such as Epstein-Barr virus, EBV, or the human herpes virus type 6, HHV6, both epidemiologically associated with multiple sclerosis. The immunological and neuropathogenic effect induced by the HERV-W protein, or WM, in vitro and in vivo are also well documented and consistent with the pathogenesis of multiple sclerosis or neurobiological dysfunction in psychosis, for example. This slide now illustrates how various infectious agents, mostly viruses, were shown to activate HERV-W and to induce a non-physiological production of its envelope protein when infecting certain cell types or when only triggering receptors at their surface. Once activated by an infectious agent, this expression may continue through paracrine activation or intercell transmission. As also illustrated here, many research groups have contributed to a better understanding of the specific receptors or signaling pathways which are triggered by particular viruses, such as the few herpes viruses given as examples here. There has also been outstanding research to understand the pathogenic relevance of HERV-W in neurological and autoimmune diseases. This is illustrated here for multiple sclerosis, in which WF activates the TLR4 pathway in microglia, which induces its aggressive phenotype, and in oligodendrocyte precursor cells, which inhibits remyelination. Neuroinflammation driven by microglia and hampered remyelination are understood to be key drivers of the neurodegeneration process, leading to disability buildup in MS. So after that, a self-sustained feedback loop of HERV-W expression in microglia was also understood to be consistent with paracrine progression causing lesion expansion, as can be seen on the bottom left. The extensive research recently made on SARS-CoV-2 also showed that this virus can trigger the expression of this immune and neuropathogenic HERV protein, which may impact both acute and post-infectious COVID-19. I will first show the in vitro evidence. To experimentally address this question, we incubated fresh peripheral blood mononuclear cells, PBMCs, from healthy blood donors in culture wells with or without infectious SARS-CoV-2 virus. On the top left, we can see that 2 hours after exposure to SARS-CoV-2, about 1/3 of the PBMC from donors showed an increase in HERV-W RNA, and also in another HERV family, HERVCOV. The graph below shows that the kinetics of cytokine release, here IL-6, occur later and cannot explain this activation. On the right panel, presenting the PBMC at 48 hours after exposure to SARS-CoV-2, specific monoclonals labeled HERV-W envelope protein in green and should label SARS-CoV-2 nucleocapsid protein in red, as in infected viral cells that are not shown here. And we can observe that on the left side, the controls without SARS-CoV virus are all negative. 2, the PBMC exposed to SARS-CoV-2 in the next column show no replication nor infection by this virus, for it should be in red and it's not seen. 3, no HERVCOV protein is seen. It should be in green in the top right pictures. And despite RNA expression, we see no protein. 4, the HERV-W envelope protein in the second column is seen in lymphoid cells, and with magnifications can be seen strongly expressed in lymphoid cells and macrophages. But the 5th point is that this was observed only in PBMC from about 25% of healthy donors, as shown in a negative culture in F' slide picture. So this recapitulates data from in vitro studies that evidence that SARS-CoV-2 can induce HERV-W expression in PBMC in about 1 in 4 healthy donors, which was confirmed in patients. We established collaborations with a university hospital in Rome, Italy, in Saragossa, Spain, and Lyon, France, to analyze the potential HERV-W activation in the blood of COVID-19 patients. In the left panel, HERV-W detection in COVID-19 patients during the first pandemic wave in Rome shows that the percentage of positive lymphocytes was very high in severe patients who were hospitalized in ICU and needed oxygen supply. The percentage of positive lymphocytes significantly increased with disease severity. And in the right panel, independent studies were performed using immunocapillary detection of the WF protein in the serum. COVID-19 patients from Spain at PCR diagnosis are presented on the left and showed positive antigenemia in about 20% of cases. On the right graph, COVID-19 patients from Lyon, France, show the increasing percentage of WF positivity in the serum with disease severity, reaching 100% of positive cases in ICU hospitalized severe ones. This was confirmed with other cohorts studied during the pandemic waves with successive variants as described in our recent publication indicated below the graphs. In the same publication, we presented immunohistochemical analysis of COVID-19 tissues from various organs. As shown here in lung epithelium, heart muscles, and intestine villi, SARS-CoV-2 infection is seen in lung pneumocytes, but HERV-W is expressed in other cell types, dominantly seen as lymphoid cells in a subepithelial region or mucosa-associated lymphoid tissue in the intestine. Also frequently in endothelial cell walls of blood vessels, as seen in the cardiac muscle here, and also occasionally in blood clots. We also observed expression of the HERV-W envelope protein in brain microglia in the absence of any SARS-CoV-2 detection. And I'm showing those pictures because the same observation was made by colleagues at the NIH with a specific and strong detection in microglia as seen on the right picture with HERV-specific monoclonal antibody, and again in the absence of SARS-CoV-2 detection. Interestingly, HERV-W expression in microglia was associated with the neurodegenerative aspects in MS as previously presented. So, since the HERV-W protein is pathogenic and has no physiological function, we explored its possible persisting expression in patients with post-COVID disorders. Given its known effect, as previously presented with few examples, this also raised the question of its contribution to some of the post-COVID symptoms. We set up for the purpose first collaboration with the post-COVID unit previously used to follow patients with chronic fatigue syndrome in Valencia, Spain. The graph on the left shows that in the pre-pandemic sera, no controls, and only 2 CFS cases had positive WF antigenemia in serum, while about 60% of COVID cases, 7 in 12 here, were positive, even more than the parallel control parallel cohorts of mixed cases with acute COVID-19, showing about 40% of positives, 9 in 22. In parallel, we collaborated with the psychiatry department of Paris Créteil Hospital in France, where Professor Le Boyer raised the question of an increased number of cases with depressive or even psychotic disorders in new cases who did not have a severe form of COVID-19 and in psychotic patients with new acute episodes after possibly asymptomatic or unnoticed infection. In the latter case, the question also related to the observed low incidence of symptomatic COVID-19 cases due to antiviral effects of some antipsychotic drugs they were taking. As shown in the pie chart, as shown in the pie chart, only about 1/3 of the hospitalized patients with psychotic spectrum disorder, PSD, had WF antigen in the serum, but as shown in the graph below, these were almost exclusively observed among patients with a positive SARS-CoV-2 serology. These observations altogether confirm the persisting HERV-W expression in a significant number of post-COVID cases, but from very different cohorts. So we analyzed another cohort of patients with post-COVID neurocognitive impairment from the Northwestern Hospital clinics in Chicago, USA, in collaboration with Professor Igor Koralnik. Here too, a significant number of patients with long COVID, or here called post-acute sequelae of COVID, PASC, had positive WF antigenemia shown on the top left graph. However, while they represented about 1/3 of all PASC cases, nearly 70% of them were positive among cases who had required hospitalization during the acute phase of COVID-19 infection. Moreover, as seen in the graph below, the WF positive cases had significantly lower post-acute duration of symptoms than the WF negative subgroup. And on the top right, we considered the SARS-CoV-2 serology and saw no significant difference from healthy control for anti-spike protein, but we saw a significant difference for the IgG against the nucleocapsids from SARS-CoV-2. And interestingly, most anti-IgN, IgG positive samples overlapped with those positive for WF antigenemia. On the graphs below, the left one showing IL-6 detection indicated that IL-6 was significantly more often elevated in WF positive patients, but more strikingly on the right, lower right graph, the dosage of serum neurofilament light chain with corrected Z-score for H showed 100% elevated values above the normal baseline within the WF positive subgroup. We should be here reminded that NFL has now been shown a very robust biomarker of neurodegeneration. So these cohorts again confirm the detection in long COVID patients. And here it must be highlighted that long COVID is presently and most likely for years to come a serious public health issue. The latest UK report on long COVID shows that about 2 million of affected persons in the UK can be seen in the recent reports, and importantly, that a high proportion of those patients are becoming chronically ill. Other reports show that more than 17 million are affected in Europe and from 16 to 34 million in the USA based on CDC estimates. In this context, it appeared relevant to propose a therapeutic avenue for long COVID patients positive for WM. The difficulty of running trials in long COVID is the heterogeneity of the patient's population in many aspects, in terms of symptoms that are numerous and often overlapping. The strength of the approach based on the HERV biology is to be able to select a population of homogeneous patients based on HERV-W positive antigenemia. We have thus set up an immunocapillary assay, also called digital Western blot, for the identification of long COVID patients with positive WF antigenemia, as shown in different cohorts in the previous slides. And therefore, we can target a homogeneous population positive to WF. In long COVID, the principle of precision medicine must be and can be applied, and this is what we have done. So GeNeuro has developed a humanized IgG4 antibody, neutralizing the pathogenic effects of HERV-W, which was already tested in phase 2 clinical trials with over 450 patient years of experience, showing a positive effect by MRI on neurodegeneration and myelin preservation, and importantly, an excellent safety profile. Temelimab was ready for use and has now been approved for post-COVID trial in Switzerland, Italy, and Spain. The presently ongoing phase 2 trial is based on the pre-screening of patients with long COVID for HERV-W protein detection in serum to ensure that only patients who may benefit from the treatment are included, as described in the top of the slides, of this slide. Patients are then randomized with 1-to-1 ratio between the groups treated with temelimab or with placebo. And the objective is to include 200 patients and to evaluate the effects on cognition and fatigue. To date, 5 sites are actively recruiting in Switzerland, 3 in Spain, and 2 in Italy, with several more expected in the next few weeks. We can already report that in a real-life clinical setting, over 25% of the patients have positive WF antigenemia. The top-line results from this HERV biomarker-driven clinical trial in long COVID are expected in between the first and the second quarters of 2024. Well, my last slide will be to acknowledge the multiple group from Switzerland, France, Italy, Germany, Spain, Mexico, and the USA, with whom we have collaborated on different studies from which I have presented only some results in this presentation. Thank you very much.
Thank you very much, Hervé. That was really great, very, very stimulating, and very, very interesting. And congratulations also to you and the colleagues and the company for long-term effort. We know where it comes from. And of course, we have the name of Christophe Mérieux also at the beginning of all this adventure. So thank you very much. So we have, as always, the possibility to ask questions in the Q&A. You have already a first question from Bob Gallo. I don't know if you can see it, otherwise I can read it for you.
Yes, please, because I don't see it. And
okay. So from Bob Gallo, thank you for this superb and for me highly instructive presentation. I was not clear on the induction of HERV-W in uninfected by SARS-2 brain cells. Do you say or think this is from a SARS-2 protein like the spike, or perhaps a cytokine?
It's a big question because we think from what we learned in MS, which is the situation that we have the best studied, is that there is a probable initial activation in the periphery of the central nervous system. And this may take place in the leptomeninges, and leptomeningitis can be seen in MS and might be seen also in COVID-19 if we looked at it. And it appears to have some non-productive infection in macrophages or microglia, because herpes viruses or SARS-CoV-2 might be engulfed and by antigen-presenting cells, but meanwhile trigger the expression of this endogenous retrovirus. In the slide where I quickly presented the pathway, the receptors that have been identified for different viruses and not yet identified for SARS-CoV-2, I showed that the stimulation does not require replication, but an initial stimulation in few cells in the CNS initiates what we later saw to be autocrine and paracrine activation mediated by the ENF protein itself. So I think that it might be the secreted form of this envelope protein that we have also identified and that we detect in the serum that may have some paracrine effects.
Thank you very much. To follow up on this, I have a question which is, we do not see you again. Okay, I have a question which is obvious, in fact, but did you notice any difference in the level of expression of HERV-W envelope depending on the variants of SARS-CoV-2? I mean, is there any difference, for example, with the Omicron? Because we all know that SARS long COVID seems to be, the prevalence seems to be really significantly lower in patients infected by the Omicron and Omicron-derived variants.
Yes, I could not show everything, and in the recently published paper in iScience, we have shown that the different variants did trigger this expression, and we saw it in patients with COVID-19 also. So the answer is yes. We should compare with in vitro studies relative potency of this activation, but we now believe it's receptor mediated, and it may not be an issue.
Thank you. And finally, the detection in the serum, sorry, I may have missed this, is do you have now an ELISA which has been set up for the detection in the serum?
It's not an ELISA. All attempts to set up an ELISA, meaning to have a capture in aqueous phase, failed. We described in a previous paper the physicochemical and biochemical properties of this antigen, and the conclusion was that if we didn't capture it physically, as in a Western blot, we would lose it. So we had the possibility to use the platform from Protein Simple that allows immunocapillary digital Western blot analysis on very small volumes of sample and with high accuracy and sensitivity. And yes, we could use this platform as an automated immunoassay detection, but not ELISA.
Thank you very much. I think that we are coming at the end, but I have just a final comment. We have at the Global Virus Network has organized from the very beginning of the COVID-19 pandemics, a task force, which has been meeting at regular time interval. Now, of course, the meetings are much more interspersed because COVID-19 has slowed down, but we have a specific task force on long COVID. So I think that Alfredo Garzino, who is our program director, may contact you, and you might be interested to be part of the long COVID task force. And that will allow also maybe more direct interactions on your findings than today, you see, or where we have the general GVN audience. Anyway, I do not see other questions, and generally we want to have relatively this type of one-shot presentation on new findings. This has been clearly the case. Thank you so much, Hervé, very interesting. Thank you all for attending. And as always, we will welcome you for the next webinars. Thank you.
Thank you very much. And to all attendees also, thank you.
Bye.
And